Hello, so today we're talking about three main things, environmental cultures, transmission, and hand washing. I do have a little bit more to go through, so I'm going to jump right in. So the first thing we're going to go over is environmental cultures. I picked to swab my mouse. It's a gaming mouse, World of Warcraft gaming mouse, but it's a normal standard gaming mouse. Everyone picked something different. A few people picked different dollar bills. A few people picked coins, shoes, drinking fountains, door handles, cell phone cases. Someone actually brought a swab home, sampled their chicken coop water. It was really just a variety, whatever people really wanted to do. So that's really, really cool. I picked my mouse, so we're actually going to talk about that next, what we did next. So yeah, moving on to that part. What we then did is after we got done swabbing it, we used a swab for it's not this is the normal normal way to streak. So we didn't use a flat pepper loop, we actually used a swab in this. But as you see, you just make motions, and I'm going to use a different color, and I'm going to kind of, please, excuse my drawing because that is on a computer. So you make one line straight down, and then you take the swab and you go back and forth and back and forth. And you want to fill in as much as you can without ever going back up. You only want to ever move down. So you keep on going and you really, really, really fill that up. That is where you'll be getting all of your bacteria, all of your growth from, because after that you're going to take a loop. We took a loop, we incinerated it. We have incinerators there. You keep an incinerator until it is actually orange glowing hot. You then let it cool off for 15 to 20, 25 seconds, depending, on how long you left it in there after sort of glow orange hot. And then after that, because if you use that, you will be, number one, melting the agar when it's that warm, but you're also be killing all the bacteria that's going on. They're going to be killing every single viable thing that you have put on that plate. So you really, really don't, if you use it too warm, it really just messes everything up. So then you go into quadrant 2, and I'm going to turn off the camera. So then you go into quadrant 2, and you go back, you start in quadrant 1, and then you come down. And then you go up and down some. These lines will be a little bit farther spaced apart, but they're still pretty close. You never want to hit back up into previous parts, because you never want to have, like, okay, for example, I would not want this line to go up into like that line. That's a no. You really want to just, because you're trying to space it out to open it up so you only have one type colony. Most of the time when you do this, you have a lot of organism on this part. So if you keep on going back up, you will not be able to get those single colonies. And that's what you really need to be able to put it onto a glass slide to look underneath in the microscope. By this time, you're actually using a loop. You're not any longer. You're using a loop. You're not. We would throw away the swab after the first quadrant. The loop, you use the tip of it, so it's pretty much the size of a pencil, like pencil lead. It's very, very thin, very, very, very thin, to make sure you're not spreading too much. So quadrant 3, once again, you kind of do the same thing. Once you get out of quadrant 2, you never go back into quadrant 1. So you, once again, kind of take it out. You want to go back into quadrant 2 to be able to get that organism, but it's not as much anymore. So that would be quadrant 3. Once again, your lines will be a little bit closer together. I do apologize. It's a little bit tricky to do this on the computer. So then in quadrant 4, this is where you'll really be getting the individualized, just the colonies of organism. Normally you get it right in that area. Every once in a while you'll get over here, but normally it's right in there. So that is how one would streak. So yes, I streak in class, but that's just kind of the basics of how someone would streak on a plate. So we use a blood agar plate. This is a picture of a plain, empty, never used blood agar plate. They call it a blood agar plate because it's actually agar media, but then it is enriched with 5% sheep's blood. Each agar plate kind of varies. It really depends on what company you order through and what you want. Every single one that we use in the lab is enriched with sheep's blood. So that's what you're missing up here. So this is my plate from the mouse. As you can see, a few things grew. I have one colony right there, one more colony right there. Then that's a colony itself, and that's a colony itself. That's a colony. One unique thing about that is you can kind of see how it has like that outer rim. That is very unique. Not many have that. You can see a few more colonies in that area, one right there. So I mean, a few things grew, not much, but definitely a few things grew. So that was kind of cool to find out. So then after that, you put it on a slide. To not take too much time on this. So each slide, there's 4 main steps to it. You first fix it, you let it completely dry, dip it in methanol, and then you take that out, let it dry again, and then you put crystal violet dye on it. So as you can see right there, click crystal violet dye, and then that will turn it purple. So if you look underneath the microscope and you have something purple, that's a gram-positive. After that, you use the mordant, and then you decolorize it. The mordant that we use is iodine. Then you decolorize, and then after that you use saffron. And then it's a red color, but it really shows up as pink, so it's pinkish red. But if you see that, then it's gram-negative. And this actually I'll do to their cell membranes. So this is a gram-negative, so that one will actually here. So this is gram-negative. So we're actually going to use a highlighter because I feel like that's going to be easier. So gram-negative is a pink red. And then I'm going to turn off the camera to do this just a little bit better. And then gram-positive is purple. So the gram-positive has a thicker wall. So when you put that purple dye in it, it goes all the way down in that peptoglycogen. And it stays there. That's what, so when you use that, mordant, it really just locks it in there. Now for the gram-positive, when you put that purple in there, it can only go a little ways. It can only go to right there because that is their, using white does not work well. Sorry about that. So you use that and it can only go down so far. So it can't really get sealed in for that exact reason because there's nowhere for it to go. So when you put the iodine on and then you put the, decolorizer on, that kind of takes away that first layer. So now you can really start to lock it in down there in the peptoglycogen. And that is how it has 2 different colors, which is kind of neat. So this is the slide. This is what I got on my slide. And this is a gram-positive cocci. So the picture's a bit blurry. But as you can see, kind of, it's tiny, tiny circles. Now, this is 1,000 times what a human eye can see. So it's it's in the grand scheme of life, it's not very far zoomed in, but on the normal, what most people ever see in their life, this is a lot farther than they ever see. So that's really neat. So things that had the most growth, you're probably wondering, like, what on earth grew the most? Like, that's kind of scary. The stuff is, it's yikes. So the things that grew the most, drinking fountains are really just a very amazing home for any microorganism. Water is required for anything to grow. Moisture is. So anything with moisture will grow a ton. Shoes are really bad. There's a lot of organism on the ground. Shoes get brought place to place to place. Have you ever washed your shoes? I'm guessing that would be a no. Shoes are very bad. Very, very bad. Money had a few things. One of the other things is that one thing that a lot of people found interesting is that a door and your cell phone case have about the same amount of growth on them. And people are like, why? Like, that should not be happening, not making people touch my phone. So think about this. When you open up a door, let's say that you open up a door, you have your phone in your hand or you do whatever. Even if you wash your hands after you touch the door, do you touch your phone in between that? Now, something for you to think on about your personal life. Let's say that you do wash your hands before dinner. Do you touch your phone in between them? Do you touch your shoes? Do you touch any money? Do you wipe the spots that you don't wipe down? Maybe a countertop that on the very edge where you know where something sits. Maybe a drawer handle. Do you touch those before you set the table? If not, yeah, I'm eliminating some, but that organism that you touched your phone and then you emptied out the dishwasher, do you wash your hands before you empty out the dishwasher? So just a few things for individuals to think on. So one of the things that is kind of really neat, only gram-positive bacilli have this. So you can see it is right down here more of, you can see it's purple. That means it's positive. So gram-positive bacilli, they can have endospores, but you can kind of see that little opening right there. Endospores actually make it so it's a mode of survival. It will be then that they actually, before they die, they create that endospore, and then they kind of duplicate. It's not reproduction. It's not this. It is purely a mode of survival. Endospores are pretty rare. They're not seen very often, but I just thought I'd kind of throw that in because they are really, really, really cool. So the next thing we're going to talk on is transmission. So I'm going to jump right in again because this one is a lot of fun to talk about. So do you think, a few semesters ago, my instructor, Mary, she had quite a few sick people in her class that they just had a cold. So she gave them agar plates, and she gave a healthy student an agar plate, and whenever the sick person would cough, then the healthy person would chew it right into the plate. So the question is, do you think that someone who has a cold will cough up more bacteria onto that plate than someone out of the mouth of a healthy person? So respiratory infections spread. Airborne transmissions are a major source of infection. That we all know that because they're in the air, you can breathe it right in. You don't have to touch anything. You don't have to do this. You just have to be in the same room of someone else who had maybe even TB within hours. Honestly, it lasts that certain one lasts in the air for quite a few hours. But so airborne is normally a huge concern, but even respiratory, which is more of it's by not water, but it's not an airborne disease. As you can see, it's spread by mucus more than anything else. So this individual coughed 7 times. As you can see, they were quite sick. They were probably in the peak of the sickness right before they started to get healthier. This person coughed 7 times, and you can see this is, remember, it's supposed to be a plain red plate. The healthy individual also coughed 7 times, and you can see there is a little growth there, a little growth there, a little tiny thing there, but that is it. That is it. This is the healthy individual. So yeah, there's still some. This individual coughed 32 times. Once again, it's pretty, and you can also see how the cough kind of spreads out, because they coughed right in the center, but as you can see, it does spread out. So when you do cough, make sure you cough into a sleeve or something, because yeah, it spreads out. You really do just spread the infection. This is a healthy individual that coughed 32 times. They have a tiny little growth right there, and that is it. This person coughed 10 times. That was the healthy individual, 10 times. They had nothing on there at all, which is quite amazing. So this is in the air. So my instructor then put this one down in the air. This is just what was growing in the air. She left the agar plate open. So it's just literally what was floating around in the environment. And this is what grew. So airborne transmissions, that's why they're a little bit more of a scary disease if they're a very killer one, because they do spread so easily. And there is stuff just hanging around in our area, as you can see. And this is all bacteria. This is not even molds or anything like that. Now, this one is fungal and bacteria and mold. So you can see, like, that's a mold right there. So you can definitely see that. But overall, it's just, there is stuff in the air. And it does help us build up our immunity. But yeah, kind of interesting. So now the other thing on transmission is, do you think that if someone, let's say that someone coughed into their hands, or they touched something that was filled with, bacteria, do you think shaking hands, or do you think passing the, okay, so let's say that this water bottle, I have, let's say, tuberculosis. That's a common known one. It's quite deadly. Let's say I licked the water bottle, okay? Let's say, actually TB is a bad one because that's an airborne disease. But let's just say that it spreads, mainly by contact. So if this bottle was covered in a disease, do you think passing the bottle or me touching it and then shaking hands with someone would spread more? So we actually, this is Staph aureus, which is, whatever. So there is types of transmission. I'm not going to spend too long on this, but person to person is called direct, Indirect is a use of a fomite. So what happened is that, so these were the transmission plates. So a tube contains Staph aureus, and that it was put all along a tube on the outside. So she coated a tube with Staph aureus, the outside of the tube. So the first student who touched the tube and initially shook hands with others, that was the source of infection. There are two sides, the people who pass the tube then, and then handshakers. So the tube are passed from the person to person, and then they cultured their handprints that came into contact with the tube, and then the handshakers shook hands and then cultured their prints of, the people that, I know, the handshakers, yeah, they cultured their hand that got shook. Everyone was wearing gloves, so this was a safe activity. And then make a prediction what you think is going to happen if shaking hands or passing the tube will produce more, spreading of this. So fomite is an inanimate, non-living object. So that would be the tube, since it's just a little, test tube like that. But it's just an inanimate object, so that would be a fomite. So when it's handled properly, healthcare's who passes stuff, and when they don't do it properly, when it's not done correctly, they can really spread a ton, and you will kind of see that. So a reservoir is on any site where a pathogen can multiply or have it survive. So that would be, if you have something, if a sick person touched a pen and then you touch a pen and touch your mouth, the pen would have been the reservoir. It could also be a person. Let's say that I, on my skin, had a infectious disease and I somehow got it, I don't know, I brushed a piece of hair, out of my dog's, like I moved her fur. If she then got that sickness, I would have been in the reservoir. So reservoir can be anything. It can be living people, it can be inanimate objects, it can be materials, it can be anything. So this was a source. These are huge plates. That's the handprint, and you can kind of see, there's a finger, there's a finger, there's a finger, there's a finger. Then that, will be the palm, where the palm is. So first we're going to the handshaker. So this is the first handshaker. So you can see that they still had quite a bit of an organism left on them. Handshaker 2, that is where the palm would have been. Handshaker 3 got a really nice handprint. You can really see the nice outlines of the fingers right there and right there, right there. You can even see some of the palm. Handshaker 4 had a little bit. Handshaker 5 had a little. 6, it was pretty much gone. 7, you just had dots kind of around. And then 8, you can see a fingerprint, but that's pretty much it. There's some there and some, all around there. But really the main thing in here is that fingerprint. Handshaker 9, in this procedure, she had 18 students. One of the students actually never returned the plate. So that is very interesting. So this is a source again, same source. And then this is a tube. So that's the tube. This is a handprint of the the people who touched to grab the tube. So this is the second person who held onto the tube. Third, fourth, fifth, sixth, seventh, and by now on the 8th one they only had a little bit. This is the that's the palm. As you can see, there really is a lot more growth when it comes to passing the object. If the object's the original thing, that's what has the most. So if you pass that has that, it really, really, really is going to give more and more bacteria or organism onto the person who touches it rather than just a mere handshake. So the next one is hand washing, and does that actually help? I'm actually taking a little drink right now because I've been talking a lot. Okay. So hand washing, we actually did hand washing hand washing lab. So what we did is this was our auger plate, you could see. So we had like a little line down the middle, and then a before side, after side. So what we did is we selected a soap. We actually before that we took a swab, dipped it in sterile water, swabbed around our hand. So, wherever you want to, you just took the swabbed all around your hand. People were moving around jewelry and doing this kind of stuff. And then you put that swab, you would streak it down the middle line and then side to side. And then you washed your hands However, you may do that normally, whatever you think would be an efficient, an effective wash, and then that exact same thing, the exact same way, you swabbed it on the after side. So, what do you think? Do you think there'll be more growth before or after? So this is my plate. So whenever you make a plate, you have to, wrong color. Whenever you make a plate, you have to put the date, what it was, so HW. So like if you do, Staphylococcus aureus, you do SA. If you do, gosh, let me think. If you do like, I mean, just, yeah. So, HW hand washing. Environmental cultures, we put environment. So this, is flipped down here. So this is the before side, unlike up here, because, you turn this over. And then this is the after side. As you can see, there's a little bit more growth after. And people go, how can you have more growth after you wash your hands? How does that make sense? Well, what it is when you wash your hands, the top 2 layers of your skin is mainly dead skin cells. So when you wash your hands with that friction, you really shed a lot of those skin cells. So you actually have your own microflora coming up. So when that happens, this is your natural. It's not harmful. It's not this to every single individual. Like my own flora, my normal flora on my hands when I do that. My own personal flora is not going to hurt me. So that's actually why there's more growth afterwards. But yeah, even before, I did not have that much. We actually had a lab exam before that, so my hands were in gloves throughout that because we were picking stuff up. So I washed my hands about an hour before. I had gloves on, so that's why I didn't have much growth, because my hands were protected the whole entire for an hour beforehand, and I washed my hands before that. But there's normally more growth you see afterwards because you're moving all that your own flora up when you wash your hands, but all the harmful stuff that gets washed off. So one of my friends, she actually washed her hands, touched the sink to turn it off, touched the towel dispenser to, pump out the towel, dried her hands, and then she went and then she did it. Then she swabbed her hand, the after side. If you look at this, if you look at the before side, you can kind of see how, you can see like the dark white parts like right there in the middle, but you can also see how there's like clearing. Like right in there, you can kind of see it's just like a little bit of a different color than the outside red. You can really see it like right there, how you can see that line. That is called hemolysis. That is where they actually either partially or completely destroy the red blood cells, because it is sheep's blood, so it's either full, complete, or partial destruction of red blood cells. That is not part of anyone that's even semi-healthy normal flora. That is not a good thing. That is actually what strep throat does. Streptococcus pyogenes, it actually eats at the throat, and that's why you get some white patches. It's because it's eating the blood. It's destroying that blood. So this is the before side. And you can see there is quite a bit more of normal flora on her side. Quite a few more, not even normal flora, but just in general, normal flora, bacteria, really anything. But if you look at, this is in the after side. If you look, you see that. You see the hemolysis all around in here. That is not normal flora. That is what she picked up from retouching the sink, retouching that towel dispenser. Oh, that's a little bit off. Retouching that towel dispenser. That is what she picked back up. That is not good. That is not a healthy thing that you want in your body. Just because they do have hemolysis does not mean that it is strep throat. In fact, many different things are able to be alpha or beta. Hemolysis beta is complete destruction. Alpha is partial. Gamma hemolysis is no destruction. But it does not have to be strep throat to be able to destroy or partially destroy red blood cells. But that is not part of any healthy at all normal flora. So when you touch that, it really does take away all the work that you just did of washing, not all the work, but a good majority of the work that you just did washing your hands, because you're re-picking up every single thing that you just washed off. Because you touched that sink beforehand, you touched that, if someone else didn't properly wash your hands, they touched that towel dispenser, if you touch the door, it's really just taking away a lot of what you just did. So when they say, use a towel to turn off the sink, or to, even pump the towel dispenser before with your elbow, or even the door, use a paper towel, that is why. I don't know how many of you guys know what a CNA is, but a CNA is a certified nursing assistant. You actually have to go through a 3-credit college course. It is half of the, so it's a 16-week class. The first half of the class will be pure book. The second half will be clinicals. I actually did mine over the summer, so the first month and a half was book. Second month, well, the second the second half of the second month and then the third month, that was clinical for me. Um, then after you get done and you get 72% or above in that class and you've officially passed, you then have to take a actual Wisconsin State exam. It's called the Pearson View. There is a written portion and there is a practical portion where you actually have to do. You get a sheet of paper. You do not know what's going to be on there. There's going to be 3 tasks. Um, Before though, you have to wash your hands. If you do not wash your hands long enough, if you touch the sink at any point, you have to acknowledge, oops, I touched the sink. I'm gonna I'm gonna rerun the water on my hands, get new soap, and re-scrub my hands. Once you touch that sink, you have to start all the way over. Because other people don't do that. That sink is not sterile. That sink is not clean. That sink has all of this crap. All of this crap is just floating around in that sink. So when you touch that stuff, when you touch the sink, when you touch the faucet, when you touch the towel dispenser, when you touch the door, even if you put your purse on the counter, grab your purse. If you touch that bottom of your purse, you're practically touching the counter. So it really is. When you wash your hands, you want to make sure you get a good washing. You want to make sure that you're not touching what you touched before. It really is important to do that because you just take away all the work. And then with that, that is actually the end of my presentation. I really do appreciate you guys listening. I had a wonderful time making this. I think this is all very fascinating. Um, I plan on touching base a little bit more with this stuff later, but I hope you guys enjoyed listening and watching as much as I did making it. Thank you.